titanium as one of these chemicals that sort of was by accident discovered to have very good refractive properties. And by refractive properties, we mean that it has very good light scattering ability. So the largest use of titanium dioxide is as a pigment. And it was actually, we go back over about 105 years ago, that it was actually discovered how to manufacture titanium dioxide for use as a pigment. And it's one of these things that you read about and figured that, well, there can't be a lot of uses for titanium dioxide. However, since its inception as being able to be manufactured from titanium ore, there's been over 200 million tons of titanium dioxide produced, with probably about 90% of that titanium dioxide being used as a pigment. So it's used as a pigment, that anything that requires a white color in a number of products. So these are paints and plastics, enamels, paper products. It also has very large uses in personal care products, such as toothpaste and cosmetics. It's also a primary ingredient in sunscreens because it has a unique ability to absorb and refract ultraviolet rays, which would be harmful to human skin. So, but since its use or identification as a very pure white color, it also has extensive use in foods. So it's approved for use in a very large number of food categories. And in fact, because the original assessments identified no toxicity associated with its use, the use levels are according GMP, which is good manufacturing practice. So theoretically, any quantity of titanium dioxide that is deemed to be necessary for its purpose of use, which would be to impact a white color to the food, is approved. So the whole point of titanium dioxide is to brighten food products. There's also some potential use as an anti-caking process based on its granular or crystalline structure. And if you look at the wavelength of visible light, which is from 400 to 700 nanometers, the optimum wavelength of light that titanium dioxide imparts is usually between 200 and 350 nanometers, so right in the visible light range. And this allows it to impart the strong white color, in this case, to foods. So something which is unique and very applicable to titanium dioxide assessment, especially for use as foods, titanium dioxide crystals or particles less than 100 nanometers are basically transparent. So therefore, they would serve absolutely no purpose for use as a food additive, because they would not impart the white color which titanium dioxide is known for. So we can have the next slide. So as I mentioned, there's probably earlier uses of titanium dioxide before the food regulatory agencies actually caught up to industry practices of applying chemicals that had not gone through a full regulatory assessment, because titanium dioxide first entered into production back in actually 1916. So one of the earlier food regulatory agencies that approved the use of titanium dioxide was the US FDA. And not too long after that, the Joint FAO-WHO Expert Committee on Food Additives, JECFA, conducted their original assessment back in 1969. So earlier assessments of chemicals to be used as food additives were generally not as robust as they are today. So simply the limited number of data that was required for the safety assessment of titanium dioxide was mainly predicated on the fact that in some limited human absorption studies, basically no increase in titanium could be detected. And therefore, the idea or the premise came out that titanium dioxide, when added to food, was basically very poorly bioavailable. And even based on the limited toxicity studies at relatively high doses, no effects were seen. So at that point, the JECFA decision was to set an acceptable daily intake of not limited. So essentially what they meant was that there were no toxicological effects associated with its use, and therefore we had no concerns. And that type of decision allowed Codex to assign GMP to this food additives. So essentially that meant that if industry deemed it useful to use titanium dioxide in any food categories, depending if the food was, what particular category, titanium dioxide could be used. So essentially, the issue arose that a few years ago, probably about 5 years ago, this is the issue where nanoparticles started to become known, primarily thanks to analytical chemists who were able to develop the methodology to start measuring particles reliably at less than 100 nanometers. So in the time of the earliest toxicological assessments, it was estimated that less than 5% of titanium dioxide being used in food was in the nanoscale. So this is very important because at the current time, there was a greater interest from a toxicological basis of nanoparticles. So the recent decision by EFSA was actually based on a number of studies that started to occur and were publicized back after 2016. And they're in EFSA's most recent assessment, they concluded that the initial estimate where less than 5% of titanium dioxide being used in food was composed of nanoparticles. Their new assessment had indicated that there was a significantly higher percentage of titanium nanoparticles that were likely present in food-grade titanium dioxide. So, and why JECFA had decided to update its previous opinion was that in 2021, there was a new safety assessment published by Titanium Dioxide. And this was an update from their previous 2016 position where they had indicated they had no toxicological concerns regarding the use of titanium dioxide in food. So in the 2021 EFSA position, they included or considered a number of studies that were performed with pure titanium dioxide nanoparticles. And based on the results of those studies, there was some indication that titanium dioxide was involved in genotoxicity. And there's a number of studies and endpoints that they looked at but primarily it was dealing with DNA damage and chromosomal aberration. And obviously, the removal of approvals for a food additive that's used worldwide has caused some concern among other food regulatory agencies. And the majority of these who have conducted their own internal assessments subsequent to EFSA indicating they could no longer approve the safe use of titanium dioxide. And in particular, Health Canada, the Food and Drug Administration in the US, FSANZ in New Zealand and Australia, UKFSA, and more recently, JECFA, have not agreed with the EFSA opinion. And there's an asterisk by FDA because they are currently in the process of responding to a petition to ban the use of titanium dioxide, even though they have come out in support of the JECFA position. So we can go to the next slide. So as I mentioned, in the original 1969 assessment that was conducted by JECFA, they primarily relied on the fact that there were both studies on a number of species, including some human volunteers, that not analyzing for titanium dioxide, but analyzing for titanium, there was no indication of absorption from the GI tract. And generally, when you see a food additive that is not bioavailable, unless it's producing immediate site-specific effects in the GI tract, it's generally considered to be non-toxic. And if a food additive is considered to be non-toxic, JECFA usually does not assign an acceptable daily intake. They consider it to be unnecessary. And that's essentially what they indicated, was that there was no issue with having to establish an acceptable daily intake. And this is back in 1969. And because there was no issue regarding toxicity, there was no specific dietary exposure conducted at the time. And the advice to the Codex Alimentarius Commission was that titanium dioxide could be approved for use according to good manufacturing practices. So the the update to that was that the Codex Committee on Food Additives indicated in 2021 that JECFA should reevaluate titanium dioxide. And this is primarily based on the EFSA conclusion that they could no longer consider the safe use of titanium dioxide as a food additive. So from a time standpoint, JECFA reevaluated titanium dioxide at its 2023 meeting. This included a large number of toxicity studies. And during this evaluation, JECFA also considered engineered nanoparticles. So these would be specifically pure test substances produced primarily 100% of titanium nanoparticles. So all sizes of titanium dioxide less than 100 nanometers. And these were included in the evaluation that JECFA conducted. They also considered that there were sufficient studies conducted with E171. And in the Codex system, the food additive numbering scheme is INS, so International Numbering System, and that's also 171. So if you see any publication that says E171 or INS 171, these all deal with food-grade titanium dioxide. So one of the main issues that JECFA considered in looking at some of the properties of titanium dioxide is that when you have pure engineered titanium dioxide nanoparticles, so these have a very narrow size distribution range, and based on the size distribution range, they have different physical chemical properties, and they are not considered to be representative of food grade titanium dioxide or INS 171. So I think this is an important distinction, that JECFA considered that there was sufficient studies that had been conducted with E171 or INS 171 involving dietary exposure for them to conduct their assessment. But more importantly, they considered studies that had been conducted with only pure titanium dioxide nanoparticles not to be representative of what was being used in food. So we can go to the next slide. So this is just some of the INS 171 or food grade titanium dioxide particle size distribution as determined by transmission electron microscopy. So there's relatively detailed analytical methodologies that is required to get a size distribution of particles generally considered to be representative of food grade titanium dioxide. So you see that particle size, and this is based on food grade titanium dioxide that has been supplied by the Titanium Dioxide Manufacturing Association. Also, there is analysis of food grade titanium dioxide that has been deposited with FDA in terms of use as food additives, which is part of their food additive approval process. Manufacturers have to provide the material that they intend to add for food, and FDA does their own analysis. But so essentially what you see is that the black line represents sort of the mean of the particle size distribution, even though there is a fair amount of range. And if you look at the 91st percentile range distribution, you get to the average particle size in food grade titanium dioxide as being slightly less than 100 nanometers and going up to above 250 nanometers. And this is something that would be expected considering titanium dioxide particles less than 100 nanometers serve no purpose in terms of light scattering and producing a white color associated with its use as a food additive. So one of the things that the clever chemists were able to show is that the inter-lab variability is quite great. So if you compare labs, you see that's responsible for the gray area. Those are inter-laboratory comparisons, whereas the between sample variability conducted in a lab is relatively consistent. It's only when you compare lab to lab that you see some of this variability start to come out. But the other main thing that has been identified by all food regulatory agencies is that there currently is no harmonized method available for extraction and characterization of titanium dioxide particles after it's been added to food. So you see that, and once again, this is food grade titanium dioxide. The median particle size, so at the D50, which is diameter 50, ranges from about 104 up to about 160 nanometers, which is what you'd expect if you want to produce a white color. And at the lower end of the range, there's only about 11% of particles by mass that would be considered to be nanoparticles, so less than 100 nanometers. So we can go to the next slide. So as part of the JECFA assessments, they looked at available studies, once again, both in experimental animals and humans, to see how much titanium dioxide is bioavailable. And in a well-designed and conducted animal experiment, they considered that titanium dioxide from dietary exposure, they confirmed that the absorption generally is very low. So, and this very small number is based on the analytical limit of detection. Now, if you look at titanium dioxide nanoparticles, the potential bioavailability is higher. It's usually approximately about 0.02%, and in certain cases can range up to about 0.1%. But once again, JECFA's position is that studies conducted with pure titanium dioxide nanoparticles were not totally representative of food-grade titanium dioxide, or INS 171. And I think in the variety of human studies, which look for bioavailability of titanium dioxide, JECFA came to a very important conclusion, considering this food additive has been used for decades, that if it was bioavailable, you might expect to see higher concentrations of titanium when you assessed a large age category of humans. And this they did not see. So there are human studies looking at titanium levels in blood and tissues, which spanned age ranges from late 30s, early 40s, up to late 70s, early 80s. And basically, there was no consistent increase in titanium seen across these age categories. So this gave JECFA confidence that the potential bioavailability of titanium dioxide in humans was very, very low. So one of the earlier studies that had been conducted and which JECFA reevaluated was a National Cancer Institute study specifically looking at titanium dioxide. The material used was a pigment known as Unitane 0-220. And what was conducted by FDA, and the results were available to JECFA, was it was considered that this Unitane 0-220 compound was essentially equivalent to INS 171. So that meant that this study that had been conducted by NCI, where this Unitane 0-220 was added to the diet and fed to experimental animals throughout their lifespan, was applicable to the safety assessment of INS 171. And essentially what we see at very high doses, so these doses are, 2.5 grams per kilogram body weight for rats, 7.5 grams per kilogram body weight for mice, fed over a lifetime, produce no consistent toxicological effects. So JECFA also looked at the large number of mutagenicity and gene tox studies that were available specifically for INS 171, or food-grade titanium dioxide, but indicated they could provide no convincing evidence for the possible genotoxicity, specifically of food-grade INS 171, or titanium dioxide. But one of the issues that has been raised is that the current methodologies for assessing both genotoxicity and mutagenicity aren't specifically adapted for nanomaterials. So there's a current large studies going on to try to define the specific test studies that could be applicable to the assessment of nanomaterials. So I think, and we'll discuss this study even a bit more, but The one study that EFSA actually commissioned was an EOGRT study, which just stands for Extended One Generation Reproductive Toxicology Study. And EFSA had initially said earlier that this study was not available for them to review. So this study was commissioned, and the results were available both to EFSA and JECFA. And basically, what it concluded that in looking at extended exposure to titanium dioxide resulted in no developmental or reproductive effects in these animals. And these are at relatively high doses over the course of the study, so up to a gram per kilogram body weight per day over the lifespan. And what was deemed acceptable was that there were no effects on any generation for reproductive toxicity. So, as I indicated, when you have food grade titanium dioxide and it has a relatively size distribution where the particles are greater than 100 nanometers, this would have different physical chemical properties than a dosing which was made with pure titanium dioxide nanoparticles. So, and more importantly, what JECFA realized is that the methods to produce these titanium dioxide nanoparticles were inconsistent with the same manufacturing methods used to produce food grade titanium dioxide, or INS, or E171. So the other important point that JECFA raised was that any study conducted with food grade titanium dioxide, either E171 or INF171, which are essentially the same thing, should be able to adequately assess the potential toxicity of the titanium dioxide nanoparticle fraction. So, based on this, and the fact that they confirm that titanium dioxide does have a very low bioavailability, they reaffirmed their 1969 ADI of not specified. So essentially that means that the food industry can continue to use titanium dioxide at GMP. And in fact, I think the Codex General Standard for Food Additives, the GSFA, which is a numerical list of all the uses and levels that food additives have been approved for by Codex, lists over 60 categories of food that titanium dioxide continues to be approved for use in. And the other important detail is that JECFA also updated its food additive specifications. So basically, JECFA meetings deal with both the toxicological aspects, including the exposure aspects of a food additive, but they also deal with the manufacturing and chemical aspects of a food additive. And essentially, in order to be approved for use, a food additive has to follow JECFA specifications for manufacturing and purity. So essentially what JECFA did was to describe a manufacturing process which, if followed, produces titanium dioxide, which has generally a particle range between 200 and 300 nanometers. Now, in reality, the particle size distribution in food grade titanium dioxide allows for smaller particles, but once you get into issues involving agglomeration and aggregation, these particle sizes get larger. So even though the particle sizes in the pristine food additive are likely smaller and outside the 200 to 300 nanometer range. By the time they're used in food, they increase in size. So I think this next slide, if I'm not mistaken, provides an example of that. So what we see here, this is a pure engineered sample of 6 nanometer titanium dioxide nanoparticles. So as I mentioned, once you put these in any sort of solution, what you immediately see is agglomeration, which is sort of a loosely defined collection of particles forming, and then subsequent aggregation, which involves a stronger and a denser assembly of these particles. Plus, you generally tend to see some sedimentation throughout the solution. So these titanium dioxide nanoparticles are in straight phosphate buffer. So they go in as a pristine sample of crystalline titanium dioxide nanoparticles of 6 nanometers in size. And what happens is that based on aggregation and agglomeration, you see the particle size that ultimately forms when you look at the right hand, where the median diameter of the particles changes from 6 nanometers up to over 200 nanometers. So this is what's likely forming in foods when you add food grade titanium dioxide. You get these agglomerates or aggregates forming that have the pure white refractive index that you want as a food additive, but also increase the size beyond the nanoparticle range. So we can go to the next slide. So this is the this is the study that was commissioned, and it was identified by EFSA that there was no definitive reproductive toxicity study that had been conducted. So, and this has just recently been published by Battersby et al. In 2024. So the food titanium dioxide was administered in feed. So once again, diet. And this is over a number of generations. So it generally starts with the animals being exposed through sufficient reproductive cycles, and it goes into breeding to produce an F1 generation. The F1 generation continues on exposure to the test substance. They are also then bred to produce an F2 generation. And through this study, they can also look at a variety of other endpoints, such as immunotoxicity, neural behavior, but also in this particular study, they also looked at dietary exposure to titanium dioxide and its ability through transgenerational exposure to induce these aberrant crypt foci. So as I indicated, there was exposure for 10 weeks prior to mating, continued exposure all the way up to generation of F2. And what it showed, what there was no toxicity in the animals, no developmental, and no reproductive effects. So the only thing that JECFA noted was that there was a minor issue with some developmental immunotoxicity assay. However, they did not consider this to be significant enough not to assign the no observed adverse effect level at the highest dose tested. And this developmental immunotoxicity study results primarily was related to the pure response or poor response based on the positive control. So, and this is expected to be repeated. However, EFSA, in looking at the same study, indicated that while there appeared to be no effects on reproductive toxicity, they were not convinced that this study is reflective of the particle size distribution of titanium dioxide when it's added to foods. So, and this is interesting because it's also been recognized that there is no validated method to extract titanium dioxide particles after it's been added to food and do an adequate size distribution. So this is an issue that the analytical chemists are currently dealing with, because generally to extract titanium dioxide from food, there's an acid treatment and physical disruption required, which would break apart these agglomerates or aggregates that could be added. So EFSA does still have some concern that the test substance, known as E171 or food grade titanium dioxide, any experimental results with that could be representative when it's added to food, because there's been no size distribution of the E171 after its addition to food, which really is an analytical method that has to be resolved. However, the the overall outcome of this, including the results from these specific studies, is that JECFA considered that there were studies that had been conducted with E171 or INS 171, where it's primarily being added to a food matrix, which is its main use as a food additive, and considered that these were sufficient to assess any particular toxicity from titanium dioxide nanoparticles, specifically the size distribution in food grade titanium dioxide. So essentially that led JECFA to concur with their previous position that an ADI was not required for titanium dioxide. And subsequently, JECFA reported back to Codex that there was no requirement to change its current food additive approvals. And these are all listed in the Codex General Standard for Food Additives. And we can go to the last slide.